Hello everyone, and welcome to the first edition of Flipped Classroom. This is the Ancestral Fishes lecture. So the reason we're doing this week is because you have the Martin Luther King holiday on Monday, and I wanted to try out this online lecture, then you having a quiz at the end of the week format. So I'm very interested in hearing what you all think. So this week we'll be talking about ancestral fishes, and that does not mean the old women fish that you see here from SpongeBob. It means we're going to be talking about hagfish, lamprey, sturgeon, paddlefish, gars, and a few other species. Before we get started, I just want to take a chance to give you some of my background, since last week was so jam-packed we weren't really able to do that at all. I was originally born in Pennsylvania, and I moved to Georgia with my family in high school, and I continued to be in Georgia and got my bachelor's degree at the University of Georgia in Athens. After that, I worked in Idaho for a little bit, and then I went to Utah State University in Logan, Utah, to get my master's, and that's where I studied Bear Lake. Bear Lake is a large oligotrophic lake in northern Utah and southern Idaho. And I was looking at the effects of drought on habitat availability and how that influenced the Bear Lake sculpin here and the Bear Lake Bonneville cutthroat trout. With my master's, I was able to do some scuba diving for habitat surveys. I was obviously able to handle some really awesome fish, and Bear Lake also has a non-native lake trout population, which you were able to fish during their spawning season. My dissertation research is focusing on the conservation of the Yellowstone cutthroat trout population in Yellowstone Lake, and I'm working with Dr. Christopher Guy in the co-op unit on campus. The cutthroat trout population is threatened by a non-native lake trout, which was illegally introduced into Yellowstone Lake back in the late 1980s. And since then, the National Park Service has been trying to suppress the lake trout population. And the main way that they do this is through gill netting. However, in the past few years, they've been trying to look at alternative pathways to kill the lake trout, and one of those ways is to use the carcasses to kill the embryos. So what they do is, after a gill netting event during the fall season when the lake trout spawn, they use the lake trout carcasses, like pictured here, and they place them on the lake trout spawning grounds. This causes a lot of bacterial and fungal growth, which then causes the dissolved oxygen to drop on the spawning grounds, and the eggs become inviable and do not hatch. So I'm researching how the effect of putting carcasses in these new places and using this new management strategy could possibly influence the food web of Yellowstone Lake. I also had the opportunity to work with Montana Fish, Wildlife, and Parks, looking at the Fort Peck population of paddlefish, also known as the Upper Missouri paddlefish population. Working with Cody Nagel, Steve Dalby, and other biologists, there has been a 25-year historical mark recapture data set that was collected for this population. And with all of that hard work, we were able to use mark recapture models in order to estimate abundance and survival for this population. And these are important metrics because this is a harvested population. So this will hopefully help Montana Fish, Wildlife, and Parks to continue to have this sustainable fishery in the future. So now that we've gotten the intro stuff out of the way, I think it's sometimes nice to know what your instructors do and what their background is, so you can help network or ask them questions about their research if you're truly interested. So now we're going to move on to the lesson for today, we're talking about the ancestral fishes. And I very much encourage you to follow along with your handout that is available online. And if you follow this handout, I'll also be referring to the specimens that you will be needing to reference in the classroom on Tuesday. So first, we're going to be talking about the jawless fishes, also known as Ichthy, which are the hagfish and the lamprey. You'll also notice here on this tree that there are asterisks by some of the groups, and these are the ones that you are required to know for class and for any exams. So keep that in mind. Before we start through the tree, we need to briefly go over the watersheds, not necessarily of the entire United States, but more specifically of Montana. Montana is pretty unique in that it is a headwater state. So what do I mean by this? We have waters that flow towards the Arctic, the Pacific, and the Atlantic. There are 3 major drainages in Montana: the Clark Fork, which flows into the Columbia and eventually the Pacific Ocean; the Yellowstone, which flows in the Missouri River and then into the Atlantic; the Missouri River, which flows into the Mississippi and then the Atlantic; and one minor, which is St. Mary drainage, and that flows towards Saskatchewan, the Hudson Bay, and the Arctic drainage. One important perspective when learning about fish in Montana is to understand these different major drainage systems. And using clues as to where these fish are, you can figure out which drainage they are native to. Another item of note in identification, besides the distribution of a species, is the status of that species, whether it's native or non-native, and if it's federally listed under the ESA. So ESA status can be endangered or threatened. I'm sure you've all heard of this multiple times in other classes. And then Montana also has species of special concern. If you want to know more about these, I suggest you look at the Montana Field Guide. Your Montana Fishes app, which we are hoping that you use throughout this course, is a great guide to determine whether the species is endangered, threatened, of special concern, or even native and non-native. There are 86 species of fish in Montana, comprised of 21 families. These 21 families are listed here. The number of species in each family is in parentheses. I'm going to go down the first 3 families because these are ones we'll be going over today. And the reason why I'm actually saying these out loud is that I think that they do truly help hearing these words to also remember how to spell them, which is going to be important during your tests and quizzes. Spelling will count. So sturgeons are Accipinceridae, paddlefish are Polyodontidae, and gar are Lepistostidae. Finally, if you want to know more about any of these fishes, you should look at Montana Fish, Wildlife, and Parks website, which the link is right here. Additionally, anything that's on their website is now even better organized, in my opinion, and more easily accessible on that Montana Fishes app. So please, when you're in doubt, or if you're trying to use one of the printed out keys and you're becoming frustrated, look at that Montana Fishes app because that will be a huge help. So getting into our first fish of the ancestral fishes. These are the superclass Agnatha, the hagfish and lampreys. And Agnatha means jawless fishes. Here we have a hagfish, the broad-gilled hagfish to be specific. And one of the defining characteristics of hagfish is this keratinous tongue. So these teeth on their tongue are made of the same materials as our hair and nails. And focus on this up-close picture because this will be important when you're comparing this to the lamprey. Hagfish have no jaws. They are scavengers and grasp usually carcasses like whale carcasses, and they corkscrew around to tear off pieces of flesh. They don't have eyes, but they can detect prey by scent from very long distances. They live in deep ocean depths. They have just a single median nostril for water delivery to their gills, so they're not very efficient. There's now a small commercial fishery for hagfish leather off the coast of Oregon due to their very thick skin. The combination of slime and thick skin helps them osmoregulate, as they don't have a very sophisticated kidney system to do so. If you want to look at some videos of hagfish actually eating, they're down here in this YouTube section. And also, fun fact, there is a National Hagfish Day that is observed annually on the third Wednesday in October. Hagfish are considered to be the ugliest of species. This is some very, very important information to know.
At the bottom of the ocean dwells a bizarre-looking creature. A fish so ancient it has remained unchanged for 300 million years. This is the hagfish. Its velvet smooth skin lacks scales and slithers along the ocean floor. It has a skull, but no spine. Tiny holes run along the sides of its wriggling body, some for breathing and some for sliming. But its most bizarre feature is its mouth. Like something out of an alien movie. This jawless maw is made for mincing up dead bodies. Multiple rows of sharp teeth are packed on two bony plates. With its single nostril, it picks up the sweet scent of death. A feast has arrived. It has no fins, but its paddle-like tail makes light work of swimming. The hagfish latches on and its mouth goes to work. Flesh is ripped from the carcass and shoved down its toothy throat. Soon, it's a frenzy of multiple mincing mouths. And to keep other hungry onlookers at bay, the hagfish excrete copious amounts of slime into the water. A shy shark snatches one, but ends up with a mouthful of snot instead. In minutes, the hagfish will strip the carcass to bone. It may be months before they find another meal like this.
Specimen, you should be able to identify the barbels around the mouth, the gill openings, and the slime gland pores along the side of the body. Its single median nostril, used for water intake into gills, which to note is a little bit difficult to locate on our specimen. You should also notice that, unlike lampreys, hagfishes do not have eyes or an oral disc. Moving on to lampreys, Petromyzontiformes. You should read textbook section 14.2 to look at the more detailed aspects of lamprey. They can be freshwater or anadromous, anadromous being they migrate from saltwater to freshwater. They can be parasitic as well as free living, and they're not found in Montana. They undergo distinctive metamorphosis. You should look at specimen 3.2 for those larval amicetes. Specimen 3.3 is an adult specimen of a sea lamprey. You should be able to identify the gill slits, the single median nostril, the oral disc, which is the most distinctive feature, those teeth on the oral disc as well as their tongue, their dorsal fin, their eyes, which are much more developed and easier to see than the hagfish, which is a great distinguishing characteristic. On the dissected specimen 3.4, you can see that the gills are very different compared to teleost fishes that have distinct gill arches and filaments. Lampreys are found in both northern and southern hemispheres of the world, so they have quite an extensive distribution. They have gill slits along their side for respiration and an oral disc for rasping skin and sucking out blood and other bodily fluids. There is an active program in the Great Lakes to reduce lamprey where they are non-native. They use various toxicants and low-head dams to prevent their migration upstream to spawn.
Sea lamprey originally came from the oceans, and they are known to act as a parasite towards fish once fully grown. Like most invasive species that find their way into the Great Lakes, they tend to do more bad than good for the ecosystem. This created an issue for the commercial fishing due to the sea lamprey killing off large amounts of fish, which then forced the creation of a new commission.
Sea lamprey have been around in the Great Lakes since the early 1900s. The Great Lakes Fisheries Commission was formed in 1950s, and since that time we've been treating sea lamprey.
Treating the lamprey during its younger stages is the most effective way in keeping the invasive species in check. This is due to them not going into deeper waters while growing. This gives biologists in the Fish and Wildlife Services a better chance to remove the invasive lamprey from undergoing its next step in growing.
So the lamprey, the first 3 to 4 years of their life cycle, they are filter feeders. They're burrowed in the sediment of the streams, and they're just passive. They're filter feeding. Once they start to become around 100 millimeters in length, they start to go through a transformation phase.
This transformative phase within lamprey consists of the growing of a mouth, which has a jaw filled with teeth. They then use these new tools to go hunting for fish out in the Great Lakes.
They swim out to the Great Lakes then, they attach to any fish, they're non-discriminatory, and over the course of approximately 18 months, they will kill 40 pounds of fish.
The amount of dead fish could increase if the invasive species is not kept in check and removed when possible. This also could not only cause the commercial fishing to suffer, but also have a large effect on the ecosystem as a whole. For more information about sea lamprey, you can go to our website at abc10up.com. Reporting in Marquette County, I'm Dan Callahan for ABC10 and the CW5.
They have a life history similar to salmon, that they live mostly in seawater but migrate to small tributaries to spawn. Remember, this is called anadromy. They go through an elaborate metamorphosis and body shape during development. Some species are considered threatened due to their susceptibility for dams and culverts that block upstream passage. They aren't very strong swimmers and are easily blocked, so the Pacific lampreys are specifically being threatened by these dams. They are an important food source for some Native American tribes, and in some European countries, tinned lamprey are considered a delicacy.
They gave themselves up so that we could live on this world.
Role in our life and our culture.
What you call lamprey, I guess I grew up hearing all of our old people call them eels.
He was a good food source. He was easy to catch. He was plentiful. Spiritually, he's one of us.
It was part of our daily life. It was part of our growing up. We just never thought of it and maybe took it for granted that we would always have this rich natural resource available to us.
Lamprey were once very abundant in the entire Columbia Basin and tributaries of the Columbia Basin. They were known to be, their range extended all the way up into the headwaters of the Columbia, all the way in the upper Snake Basin. Now lamprey are only really present in the mid-Columbia and the lower Columbia River.
They've been here for hundreds of millions of years, but it's only going to take 100 years to wipe them out.
You want to see him? There he is. There he is. You want to meet my friend? We'll go to the dams. We pick the eels up. Take them inland where our home country is. We take them to the streams we want. We put them in the water. The rest is up to them.
So here's an illustration comparing the hagfish and the lamprey. I would take a moment here to look at these differences and compare these illustrations here to the table I'm going to be showing on the next slide. Here's a nice table showing the differences between adult lamprey and hagfishes. I think the easiest characteristic to look for are the differences in the eyes. The hagfish, you basically cannot see these on the specimen, while in the lampreys, they're very well developed. The lateral line system in hagfish is degenerate, while in the lampreys, it is well developed. In hagfish, they do not have a vertebrae, but in lampreys, they have a cartilaginous vertebrae. Finally, barbels are present on hagfish. They have 3 pairs, while lampreys do not have barbels at all. Next, we're going to move on to chondrichthyes, which are sharks, skates, rays, and chimeras. You should read textbook sections 15.1 to 15.3 to learn more about chondrichthyes. They have distinctive placoid sandpaper-like scales and various reproductive models, including egg laying by release of leathery eggs, which you can see in specimen 3.5, which is in the box. Some species bear live young, which develop in utero, which you can see in specimen 3.6. In specimen 3.6, you should be able to identify gill slits, which you should compare to a teleos like perch or trout. So comparing these is looking at the evolution of the respiratory structure. The spiracle, the water entry pore, nostrils, the heterocercal caudal fin. So remember, heterocercal caudal fin, the caudal fin is not even from the top and the bottom. Their ancestral ventilation system causes them to need buccal pumping, so they'll open and close their mouth frequently to force water over their gills, or also ram ventilation, which you'll see when sharks are swimming with their mouth open. Just the movement is enough to get enough water over their gills for respiration. So remember here, we did hagfishes, we did lampreys, sharks and rays. You'll be responsible for identification of these specimens in jars. I'm going to take a brief moment to focus on the Sarcopterygians, which are a section of this tree that you're not responsible for in terms of the handout or the test, but I do believe these are important fish in terms of evolutionary status. The Sarcopterygians are also known as lobed-finned fishes. These are coelacanths, that's how you pronounce that word, or lungfish. They're lobe-finned because they have this fatty lobe protruding out of their bodies before the fin actually starts. Or in the case of the lungfish, it's just this fatty tendril that comes off their body. Coelacanths in particular are interesting because they were thought to be the ancestral species that led towards tetrapods, so they're also known as the terrestrial fish. Here I have a nice cartoon depicting Chucky D, so Charles Darwin, embracing his inner fish. And if you want to learn more about embracing your inner fish, you can read this book, which is a journey into evolution of tetrapods in the human body. I've read it before, and it's not a total snooze if you're interested into scientific reading. The discovery of the coelacanth was quite unique. It was believed to be extinct for 66 million years, completely disappeared from the fossil record. But then it was found in 1938. It was a big discovery at the time because it was thought to be the missing link between fish and tetrapods. While that's no longer the best supported hypothesis, this is still a very interesting discovery and a very cool fish. The first specimen was found by Marjorie Latimer. She was a museum curator and at the time got it from a local fisherman. However, J.L.B. Smith was famous for first describing it, and it's thought that he largely took credit for this finding of the specimen as well as describing it. But later on, this was uncovered, and so now the coelacanth's scientific name is a nod to Marjorie Latimer. So we're moving back on to our asterisk fish. So we're going to be moving to the Actinopterygians. So these are now jawed fishes, right? We're moving further down to more modern fish, higher evolutionary status. So first we're going to be talking about the sturgeons. Sturgeons, their family is Accipinceridae. There are 23 species of sturgeon, 4 genera, and they're in the northern hemisphere. They're the most endangered fish family. They have extended, hard snouts. They have a well-developed operculum, a ventral protrusible mouth, and large bony scutes on the side, which we talked about in last week's activity. They have a heterocercal tail, which remember is similar to the sharks. They are a large river, large lake fish, and some of them are even anadromous. Remember what that anadromous word means, similar to what salmon do. In North America, we have white, green, pallid, shovelnose, Atlantic, lake, and short-nosed sturgeon. 3 of those are in Montana. The sturgeons are known for their caviar, especially the beluga sturgeon, which is from the Caspian Sea. You should read textbook section 16.6 to learn more about the sturgeons. The 3 sturgeon species in Montana can be identified by their caudal peduncle, so the shape and size of their caudal peduncle, and by the position of barbels under their snout. You should note the sturdy scutes along the side of their bodies. Look at specimens 3.7 or 3.8 under a microscope to locate their barbels, protrusible mouth that does not have teeth, and distinctive scales. Sturgeons are one of the largest freshwater fishes, are hardly sought after commercially because of their eggs or the caviar, and are one of the most endangered fish families due to overharvest and blocking of their long-distance migrations by dams. First, we have the Kootenai White River Sturgeon. This is native and on the federal endangered species list. In Montana, they're limited to the Kootenai River of the Columbia River Basin. Note their stout caudal peduncle, so talking about this thick caudal peduncle compared to the shovelnose and the pallids, and the presence of this large spherical near their eye. We do not have a preserved specimen of this species. White sturgeon are widespread in the Pacific Northwest, but this subspecies is considered unique as it's been isolated above a waterfall for a long period of time. They do find mature adults, but no successful reproduction has been documented for decades, and the population is comprised of only large old fish. This is a similar situation to pallid sturgeon, which are in eastern Montana.
We're here at Libby Dam in northwest Montana, which is an important source of hydroelectric power and also provides flood control. Normally you wouldn't see water coming over the spillway behind me, but this week in early June, the U.S. Army Corps of Engineers, in cooperation with BPA, the U.S. Fish and Wildlife Service, and states and tribes, is spilling extra water down the river in hopes of helping white sturgeon. These are enormous, endangered, long-lived fish that haven't spawned successfully in a few decades. The idea behind this spill is to recreate more natural spring runoff conditions when the river is high and lead the sturgeon to swim upriver to much more productive spawning grounds. There are biologists out in the river checking to see whether the fish are moving.
We don't know anything about what these fish need for survival. All we know is where they're spawning now is not working.
We've been monitoring river conditions and fish migration for quite a few years now, and so what the spill test is doing is approximating a better water year in the lower river here. So we're trying to create a depth that was closer to the pre-dam conditions with spill, with the habitat work that the tribe and others are bringing onto line. And the other half is we're trying to learn if our temperature management strategy at Libby Dam is going to be effective in pairing with the flow conditions. USGS has done work for almost a decade in examining the effects of flow on substrate, how the river channel changes with flow. But what the GS did was show us through their coring that the sediments downstream have probably always been sand, silt, clay, and that our flows really had very little effect on that. What we're doing now is we're closely in tune with what flows did, and that is provide depth, move substrate. provide fish spawning migration cues to make these fish migrate where we think they could be successful in their spawning.
Well, the tribe's been working for several decades on restoring the population with the aquaculture program. Basically, we're creating the next generation here at the hatchery. We've been able to have good survival of fish that we put back out into the river, but we also realize that there's need to restore the habitats. And so that takes several different actions and activities beyond flow or spill. We need to have complex habitat where you have a deepened channel and a connected floodplain that provides those ecosystem functions that are missing right now. And then also productivity is really low. Libby Dam holds back about 60% of the phosphorus. And because phosphorus is a building block for all the different trophic levels, we're adding nutrients to the system. By restoring the nutrients, we provide the food that the fish need for survival. So habitat and food and flow probably all kind of go hand in hand.
It's going to be 20,040 for the first female out of this year class to even start to reproduce. The recruitment into the wild stock is non-existent or very limited. So we're trying to maintain the genetics until they reproduce and recruit in numbers.
Pallid sturgeon are also an endangered species. We have that specimen is 3.9A. They are native and on the ESA list. They are in the lower Missouri River and the lower Yellowstone River drainages. They are now pretty rare in that range. You should note their long, slender caudal peduncle, and their barbels are about 1/3 of the distance between their snout and their mouth. The outer ones are typically 2 times longer than the length of the inner ones. You should focus on your Montana Fishes app key for this, and I also have an image coming up to better demonstrate this compared to the shovel-nosed sturgeon. So here's an interesting picture of an actual fish. You can see here this longer outer barbel and the shorter inner barbels. There are hatchery-reared pallid sturgeons stocked into the Missouri River. This one is about 2 years post-release and was in the Yellowstone River. You can tell that it's a hatchery sturgeon because this faint yellow and faint pink line on the underside of their rostrum, that is what they're using to identify the release year of this fish. Further, there's lots of research focusing on the pallid sturgeon recovery in the Missouri River. Remember, sturgeon are very heavily impacted by dams. And so one of the case studies is looking at the den zone that's hindering pallid sturgeon recovery. So this has to do with the dam reservoir continuum and the fact that there is possibly not enough oxygen behind the dam in order to have the successful hatching of pallid sturgeon.
State Northwestern energy biologists, in conjunction with Montana Fish, Wildlife, and Parks, have been looking for evidence that pallid sturgeon are spawning in the upper Missouri, Marias, and Teton rivers. Recently collected samples may provide the fish proof that the fish are spawning and indicate a significant step to the species recovery. Northwestern energy biologists have found several sturgeon eggs and larvae in the Marias Teton River systems, but lab results have yet to confirm if they are pallid sturgeon or the more common shovel-nosed sturgeon. There are currently estimated to be about 4,900 pallid sturgeon in the 200-mile stretch of the Missouri from Maroney Dam all the way down to Fort Peck. At one time, there was only about 33 adult pallid sturgeon in total in that stretch of the river. Grant Grisak, a fish biologist, with the Northwestern Energy, says that this is a big step if they are in fact spawning.
The ultimate goal is to get them to a self-sustaining population, and because they are such a long-lived fish, we started off with such low numbers. We're probably halfway to that benchmark.
And pallid sturgeon is one of the rarest and largest freshwater fish in North America. It takes around 15 years for pallid sturgeon to reach maturity, and the first stock was released into the upper Missouri in 1997.
Next, we have shovel-nosed sturgeon. Shovel-nosed sturgeon are very easy to confuse with pallids. They have a very similar native range, and they have a similar snout and skinny caudal peduncle structure. However, to tell the difference between shovel-nosed and pallid sturgeons, you need to refer to the length of the barbels as well as the positioning of the barbels. What's easiest for me to remember is that the pallid sturgeon has this almost half circle positioning of their barbels, and their barbels on the inside are about half the size of those on the outside. The shovel nose has a shorter distance to the top of their barbels, which this is a hard thing to compare without having two specimens in hand. But the better thing to look for the shovel-nosed sturgeon, if you only have one specimen in hand, is that the barbels are completely equal to their starting point, and they're about the same length. Next, we have paddlefish, family Polyodontidae. You should read textbook 16.6 in order to learn more about the paddlefish. We have specimen 3.10 in our lab. Paddlefish are native and are listed as species of special concern in Montana. The lower Missouri and the lower Yellowstone rivers have a population of paddlefish in Montana. There is also a Chinese paddlefish in the Yangtze River. They have a very unique, elongated rostrum flattened into a paddle. This paddle is thought to function as a rudder to hold position while feeding, with the mouth open in fast current. So you can see that posture here. It also has many electroreceptors for detecting weak electric fields generated by their prey. These pores, this color that you can see, this modeling, is the electroreceptors. They have many slender, long, closely spaced gill rakers, and this is because they're a planktivore. These gill rakers help them filter the water and capture the prey in their mouths. They have a spiracle present, and this elongated operculum, which could be one of the features that you use to identify this fish. They also have a very unique eye position, with it being almost exactly on the sides of their rostrum. They also have modified ganoid scales on the dorsal lobe of their caudal fin. They have a heterocircle tail, similar to Accipinceridae, and also these sharks. Paddlefish are similar in that they are prized for their caviar. So both sturgeon and paddlefish can be easily overfished due to their late age at maturation and the focus of their flesh as well as their eggs being very useful for commercial fisheries as well as recreational fisheries.
Each spring, thousands of anglers flock to the lower Missouri and Yellowstone rivers to pursue one of Montana's oldest fish.
This is a unique species. It's a fossil species. It's been around for 60 million plus years, and people get excited about them.
But due to the paddlefish's popularity and the concern for this prehistoric fish, FWP instituted a cap on the amount of paddlefish that could be harvested.
Implementation of the 500 fish harvest cap and unlimited tags being allocated, we created this atmosphere of first come, first serve type fishery, and we progressively saw our season for the harvest of paddlefish closing earlier and earlier every year.
To provide a more fair angling opportunity, anglers who now want to harvest a paddlefish on the Missouri River upstream of Fort Peck will need to apply for a permit. The hope is this lottery system will also preserve the Missouri's paddlefish population.
Biologically, what we've been seeing happen is the earlier harvest of these paddlefish, we're kind of focusing in on the earlier run. So in any way we can spread the harvest out, the better for the overall population.
Also starting this year, anglers who harvest a paddlefish anywhere in the state will be required to report that catch. I'm going to keep this one.
The mandatory reporting is going to help us get better data on this species from a harvest component.
The deadline to apply for the Missouri River paddlefish tag is March 31st. I'm Winston Greeley, out among Montana's fish, wildlife, and parks.
Finally, we have the gar, family Lepistostidae. In Montana, we have the short-nosed gar, which is here, located right below Fort Peck Reservoir. They are a species of special concern due to their restricted distribution in Montana. You'll want to read textbook section 16.7 to learn more about gar. And also be sure to look at specimen 3.11. They are native, listed as a species of special concern. They're in the lower Missouri River below Fort Peck Dam. They have an abbreviated heterocercal caudal fin because of this section right here. They have hard, non-overlapping ganoid scales and a tubular, elongated snout with many small teeth that are very arrow-like. They also have this arrow-like shape for ambushing prey. If you remember, the poster child for ambush predators are the pike, and if you looked at the back of their bodies, or if you just chopped their head off, you could be confused as to whether this was a pike or a gar, but the reason they would be different is because of these ganoid scales. But you can see that the form of this fish, right, is related to its function, being an ambush predator. They have large vascularized swim bladders that can be used for breathing air when oxygen levels are too low in the water. For comparison, you should check out specimen 3.12, which is a spotted gar from the lower Mississippi drainage. That specimen has a much more elongated snout, but shows the tooth structure of gar very well. Gar flesh is edible, and sometimes it's even available in markets, but unlike the sturgeon they resemble, their eggs are highly toxic to humans. Gar eggs are toxic because of a protein called ichthyotoxin. So that's a fun fact. Overall, you should come out of this lab with some learning outcomes, which include the ability to describe the main differences between agnathan fishes and other fishes, remembering what agnatha means. You should be able to describe the feeding and respiration characteristics of hagfish and lamprey, and the management considerations of these two groups of fishes, so the commercial hagfish operations as well as the management of native and non-native lamprey. You should be able to identify the unique features of sharks and rays and how you distinguish gender in shark skates and rays. You should be able to know how their respiration and scales differ with bony fishes. You should be able to answer the following questions about sharks. What is the largest fish in the world, and what does it feed on? What is the likely function of the hammer in hammerhead sharks? And what are the typical way that sharks reproduce? This can be found in your text. You should be able to identify all of the species that we listed here visually, list their common names, and know their family or class name. Those can be found in the bold type in your handout. You should be able to describe their general distribution in Montana, their feeding type, and when, where, and how they spawn. You should know the likely function of the paddle in the paddlefish and characteristics that paddlefish share with sharks. And finally, you should be able to describe the distinctive features of GARs. I hope you found this useful and it wasn't too bad listening. I got a fancy microphone to make it less harsh on your ears and less echoey. Please let me know what you think of this lab setup, and we might be able to do it again in February if you deem it useful. Thanks for listening, and please remember to take the quiz associated on D2L with this lab. So this will be the Ancestral Fishes quiz. That is due the Friday of Martin Luther King week. Friday of Martin Luther King week. That quiz is on D2L. You have to take that quiz on D2L. Please let me know if you have any questions. Feel free to email me. I will be in the Tuesday lab sections with the specimens out. You are required and responsible to know and be able to identify those specimens. If for some reason you're not able to pop in at any point during the Tuesday classes, we can look at those specimens the week after, or we can set up a time that we look at them specifically when there is coordination of mine and your schedules as well as openings in that room. Once again, please feel free to email me if you have any questions, and make sure you do that quiz.